this is a very important concept. From the tissue culture, one of the bit, definitely one bit is given from this area. And see, coming to the tissue culture process. So we know about the tissue culture, that is nothing but some of the cells are separated from the plant or from the animal. See, there is a different cultures are there. Animal tissue culture is there and plant tissue culture is there. First of all, what is tissue culture? Cultivation of cells. Cultivation of cells within the laboratory conditions. Production of new organisms. Production of new organisms or production of new organs. Right? So that is nothing but tissue culture. So presence of nutrients, we are cultivating the cells, we are producing the complete organism or organs. Right? That is nothing but tissue culture. So coming to the first one is the plant tissue culture, means plant cells are cultivated within the laboratory conditions, presence of nutrient media, then the new plants production takes place, that is nothing but plant tissue culture. If the animal cells are cultivated, that is nothing but animal tissue culture process. And see the basic concept for the tissue culturing process, the basic concept for the tissue culturing process, there is a totipotency. The concept is the totipotency. Sir, what is totipotency? Totipotency is different from plant cells to animal cells. I thought most of the students are well known. I said so many times, but here we have to explain. A single cell have the capacity to produce the complete organism, that is called as totipotency. A single cell have the capacity to produce complete new organism, that is called as totipotency. So totipotency, okay, very good in plant cells. Means plant cells are complete totipotent cells. Plant cells are complete totipotent cells. But animal cells are not like that. I said here, complete, especially within the plant's case, complete new organism produced. Right? And in the case of animals, here new organs only produced. Here only organs are produced, but here complete organism is produced. So this is the basic difference between the plant tissue culture and the animal tissue culture. Sir, why this one have that complete organism production? Why this one is not have? Remember, here complexity is less than complete organism production takes place. Here complexity is more. Here only organs production takes place. Sir, how here possible? Very simple. Whatever that plant sources are there, there is a stem part or the leaf part, any plant, right? The part is taken, the part is sterilization process. Sterilization is nothing but complete cleaning process. Insert it into the nutrient media. Right? The plant part is inserted. The plant part is developed into the callus. Again, within the tissue, the plant part developed into the callus after 2 to 3 weeks. The plant part is developed into the callus. This is the callus part. This is the callus. Right? After that, the callus again taken into the another tissue. Callus taken into the another tissue. Right? Again here also nutrient media is there. But here nutrient media, the phytohormones are different. Right? Here phytohormones are different. Then here there is a plant production will be takes place. Plant production will be takes place. So here what I am trying to tell, whatever that plant cells are there, the plant cells are inserted into the nutrient media, and there is a callus production which takes place. That callus again changes into another nutrient media, whole plant, a complete plant production takes place. Complete plant production takes place. Right? That is about the plant cell totipotency process. Remember here, major crucial role is for the cultivation process. One is nutrient media. Right? Next one is the phytohormones. Nutrient media and the phytohormones, these are very important. Okay? And at the same time, we have to maintain the sterilizing conditions. These are nothing but aseptic conditions also we have to maintain. And coming to the next one, animal cells are there. Within the animal cells, all cells are not produced with the organs. Very special cells are there. These are called as stem cells. These are called as stem cells. Again, the stem cells can be divided into two types. Embryonic stem cells are there. Embryonic stem cells are there. Right? Embryonic stem cells are taken into the nutrient media. And hormones are supplied. Hormones also supplied, and here produces the organs. Maybe we know. I already said when the baby delivery takes place, that time some of the cord blood stem cells are there. The cord blood stem cells are preserved within the liquid nitrogen. In the future, if the baby had any accident, in that case, the cells are activated and the cells are subjecting for the production of the new organs, like maybe the kidney is damaged, liver is damaged, or any skin part is damaged. In that case, these cells are very, very important. These cells are very, very important for the production of organs. And other one is the adult stem cells. Within our body also some of the stem cells are there. That's the reason why whatever the bone marrow is there, within the bone marrow, from the bone marrow total new blood cells production takes place. For every 120 days, the RBC cells are destroyed and new cells are produced. Sir, how the cells production takes place? Remember, there is an adult stem cells are there, right? Adult stem cells which are involved in the cultivation process, presence of nutrients and the phytohormones. Then adult stem cells, presence of nutrients. Here also organs are produced. Right? See, anyhow, the basic concept is the plant cells have the complete totipotency capacity and the animal cells not have the complete totipotency capacity. Here, complete organism is produced, but here, complete organism is not produced. So, these are the types of the tissue culturing process. Especially today we are going to discuss about the plant tissue culturing process only, because we are studying botany, right? I am teaching botany, right? We have to discuss only the plant tissue culture process, right? I will discuss about the plant tissue culture. Coming to the next topic is the steps in plant tissue culture, right? We already discussed the types of tissue culture. One is the plant tissue culture, other one is the animal tissue culture. Now we are going to discuss about the steps which are present in the plant tissue culturing process, right? See, the very first step is the preparation of nutrient media. See, any culturing process, either in animal cell culturing, either in plant cell culturing process, nutrients are required, right? The nutrients, nothing but here sucrose is required. In the case of the plants, vitamins are required, phytohormones are required, and some of the amino acids are required. All these components are required, right? So these are required for the cultivation of plant cells in a particular ratio. In a particular ratio, we are added to the nutrient media. That is nothing but preparation of nutrient media. And next one is the sterilization process. Okay. See, the nutrient media contains some of the microorganism process. The microorganisms are killed by using autoclaving technique. Right? I will explain what is autoclave and how it works. Right? And next one is the collection and sterilization of excipient. Whatever that plant source we are taken. Okay. For suppose I want to cultivate many number of banana plants. Initially, from one banana cells, I need to collect. Whatever the collected part is there, the part is considered as explant. Right? So there is a collection of explant and sterilization of explant also there. Right? And inoculation of explant. Whatever that the collected part is there, the collected part again chopped into the small pieces. Right? And the pieces are inserted into the nutrient medium. That step is nothing but inoculation of explant. Okay? And incubation. There is an incubation period is there. After that incubation period, we got the callus within the nutrient media. Again, the callus is subjecting for the plant's production. That is the next step. After that plant production, again the plants are acclimatization means hardening and acclimatization. Hardening is nothing but acclimatization. That is nothing but we cultivated plant within the tissue culture. The conditions are very favorable and very sensitive conditions. So, if the plants are directly exposed to the normal conditions, the plants will be dead. So, before plantation, the plants are kept in a particular house. Right? That is nothing but greenhouse. See, within the greenhouse, the plants are adapted to the normal conditions. The plants are adapted to the normal conditions, right? That is nothing but acclimatization or hardening of plants. So these are the steps, and coming to the first step is the preparation of nutrient media. There is in sucrose, okay, because we know the plants carbohydrates required in the form of sucrose, right? And see, in the plants the carbohydrates are also transported in the form of sucrose. Every plant will have the capacity to absorb the sucrose, right? And vitamins are required, minerals are required, amino acids are required, phytohormones are required, and agar is required. See, what is the use of these components? See, sucrose is the carbon source, vitamins activates the enzymes, minerals which are also very important for the middle lamella formation and different parts formation, like amino acids for the protein synthesis, phytohormones for the cell division process. Agar is the solidifying agent. Agar is the solidifying agent. See, this is the liquid state nutrient media. Once the agar is added after the boiling process, the nutrient media will be forms the semi-solid state. That's nothing but see, agar is the solidifying agent. Agar is the solidifying agent, right? So that is about the preparation of nutrient media. Either in the case of the plant, either in the case of the animals, definitely the nutrients are required for the cultivation purpose, right? And these nutrients are added to the one of the flask, right? And within the flask, okay, all the nutrients are added in a particular ratio. Water is also added, and there is a carpet like this. There closes that the opening part of this flask, right? After that closing process, the flask is kept inside one of the device. This is one of the machine, right? This is nothing but autoclaving unit. This is called as autoclave. This is called as autoclave. This is nothing but autoclave, right? See, whatever that autoclave is there, the autoclave is majorly involved in the sterilization process. Autoclave is involved in the sterilization of nutrient media. Sterilization of nutrient media. So how it's involved in the sterilization? How it's involved in the sterilization? Remember, whatever that nutrients are present within the flask, right? Remember, see, inside the autoclave, high pressure is maintained. High pressure is maintained. There is nothing but 15 pascals per inch. Inside the autoclave, 15 pascal per inch pressure is maintained. Exactly at 121 degrees centigrade, 15 minutes. At 121 degrees centigrade, 15 minutes. Okay, we need to do the sterilization process at 121 degrees centigrade. At 121, not 0, not 10, not 15. At 121 degrees centigrade, 15 minutes. Okay, do the sterilization process. So at that temperature, okay, within 15 minutes, whatever that bacteria cells are present inside, whatever that bacteria cells are present within the nutrient media, fungal cells are present within the nutrient media, and inside this mission, all microorganisms will be killed. All microbes will be killed. So the step is called as sterilization process. Sterilization is done by the autoclave, and especially autoclave. Here this is one of the indicator, pressure indicator. This is one of the pressure indicator. And through this one, okay, we release steam. Whatever the steam is produced, the steam is released after the autoclaving process. Right? That is about the steam nozzle, and this is one of the pressure indicator. Right? So that is about the sterilization process. After the sterilization of nutrient media, the next one is the collection and the surface sterilization of excipient. Surface sterilization of excipient. For suppose this is one of the excipient part, that is nothing but leaf part. Okay, and the leaf part is contained. The leaf part I am using for the tissue culturing process. The leaf part, one of the leaf part I am using for the tissue culturing process. So in this case, this is one of the NaOCl, sodium hypochlorite. Sodium hypochlorite. The percentage is here 0.5%, 0.5 grams, which is dissolved within 100 ml of water, that is 0.5%. So within this 0.5% of solution, okay, exactly with a very short time. See, we need to dip whatever that explant parts are there, whatever the explant parts are there, the explant pads, we need to dip within that NaOCl solution, right? Exactly less than 1 minute. Less than 1 minute. See, more than 1 minute is there. Okay, if we maintain more than 1 minute in presence of NaOCl solution, see, cells, this substance is maybe toxic to the cells. This substance is maybe toxic to the cells. Maybe the cells will be dead. Maybe the cells will be dead. Another side is the HgCl2. Mercury chloride, HgCl2. Here also 0.5%. Either HgCl2, either NaOCl. Most of the cases in the tissue culture, HgCl2 is used. And this one is also toxic. Here also less than 1 minute we need to dip that explant parts. After that dipping process, we need to separate and dry that plant parts or explant parts. Right? So, sir, what is the use of this process? For suppose this is one of the explant parts, see, this is one of the explant part, leaf part, maybe the root part, maybe the stem part. See the surface. The surface also contains sometimes some of the microorganisms. So we need to clean that microorganisms. Once, if we kill these microorganisms, then we get the canvas followed by plants. Right? So that is about that collection. After the collection process, surface sterilization of explant. So what happens if we not give the surface sterilization? Remember, whatever the nutrient media is there within the nutrient media, contamination will be generated. Maybe the fungi, maybe the other bacteria, maybe the other microbes are grown within that nutrient media along with the plant cells. Okay, the plant cells are finally destructed. To avoid that problem, we need to do the surface sterilization process. And next is the inoculation of explant. See, after the sterilization process, whatever the nutrient media is there, this is the nutrient media, this is the solidified nutrient media, we need to prepare the crosses. Because more surface area is there, more nutrients are available and more oxygen is available. See, if we prepare the test tubes with nutrient media like this, here this is the less surface area, this is the less surface area, this is the more surface area, this is the more surface area. So presence of more surface area, more nutrients are available, more oxygen is available. Right? So inoculation of explant is nothing but whatever that surface sterilized plant pot is there, the plant pot is inserted. So this is the explant part. Inserted into the nutrient media, right? So this is about that first 4 steps, and next one is that incubation of its plant, and next one is the production of plants from the callus,